This is a University of Otago podcast.
Good evening. My name is Harlene Hain, and I have the distinct pleasure of being the Vice Chancellor of this wonderful university. And it is my distinct honor to be here with you this evening. As many of you who come to these will know, this is one of the highlights of my week because it's a time where all of the busy people who are in this room have taken time out of their schedules to come and partake of the outstanding academic brilliance that is the professoriate of the University of Otago. Many times when I stand here, I remind people that being promoted to professor in this institution is a bit like becoming the Pope. It's a very, very difficult and arduous process that takes people a very long period of time. And only the best and the brightest actually achieve it. So actually being the person who stands up here this evening to give their inaugural professorial lecture, it really does indicate a huge achievement over the course of an entire career. So it is a fantastic opportunity to see people from all academic divisions coming together to spend an hour and enjoying the brilliance of Professor Catherine Day. I would now like to hand over to Professor Helen Nicholson, who is going to tell us a little bit about Catherine's performance to date.
So good evening. It's my pleasure to introduce Catherine Day to you. Catherine was brought up in Pahiatua in the Wairarapa, and coming from an agricultural background, her first interest was becoming a vet. But she was told that she wasn't strong enough. They obviously didn't know Catherine then. Instead, she went to the local school at Makamako and then went to Marston High School and then on to Massey, where she did a BSc in biochemistry and genetics, an honours, and then a PhD with John Tweedy and Ted Baker at Massey. After that, she went to the University of California in Berkeley, where she did a postdoc with Professor Tom Abler and worked on various things, including determining the structure of the tumour suppressor protein, APC. And then in 1995, she was lured back to New Zealand on a forced postdoc fellowship, and her desire then was to establish her own group, which she did, looking at protein interactions, particularly around the process of apoptosis. And she developed some collaborations then, particularly with people in Weehai, which she continues to this day. In 2001, she moved to Otago to take up a lectureship with the expectation that she would stay here just for 5 years. 12 years later, or 11 years later, thankfully she's still here. So she quickly settled into Otago. She was promoted within 2 years of her being here to senior lecturer. And since that time, she has been continuously funded, either by Marsden or HRC or both. She has published extensively in high-impact factor journals, established her group here, has graduated a good number of PhD students, and is now internationally recognised in her area of structural biology and apoptosis. As well as her numerous publications, she's been invited as an invited speaker internationally all around the world, and in 2010 was awarded the New Zealand Society for Biochemistry and Molecular Biology Life Technologies Prize. As well as being herself an active researcher and training her own group and her own students, Catherine has taken an active role in promoting science and facilitating science within New Zealand. She's been on various assessing panels, including the one of the HRC assessing panels, and certainly from the school's point of view, for the last 4 years, she's been our Associate Dean Research and has done a fantastic job developing research across the school and also helping to develop and support young and emerging researchers. So it's my great pleasure to welcome Catherine Day to give her talk, which is called Molecular Assemblies and Cell Suicide.
Okay, so thanks to Harleen and to Helen for the kind words, and thank you to all of you for taking time out of your busy schedules to come. It's really a privilege to have see so many faces here. Many I know, but a few I don't, I think. So thank you all for taking the time to attend. So I'm going to talk today about molecular assemblies, and in particular I'm going to talk about protein assemblies and how they can regulate cell survival or cell death in this case. And in particular, I'm going to talk about two examples where my group has made a contribution to our understanding of protein function, which has then enabled or helped with the development of anti-cancer compounds, which are currently in clinical trials. And so this, I guess, I'll take these two examples to represent research in my laboratory. And I guess before I start talking about the details, I'll just sort of want to step back and talk a little bit about the cell. And here I have shown a schematic, and I should thank Bronwyn Carlisle for a lot of help with the schematics that we'll see as we go through the talk. So this is a schematic of the cell, and the cell comprises many things, and in particular, many of you will know that the cell comprises a DNA or the genome, and we've heard a lot about the genome in the last 10 years, really through the pioneering work of genome sequencing projects, and we know that now that the human genome encodes 30,000 genes. That's actually far fewer genes than we expected. And really, it's surprising absence of genes, which is kind of an interesting thing about human cells, because plants have far more genes than what we do. And so really then it's meant that we've had to look in other places for the complexity of these cells. And this really, at least a lot of the complexity lies in the proteins. So as a gene is transcribed and translated, we get a linear polypeptide of amino acids, which then turn into a three-dimensional structure. And I'll come back to these three-dimensional structures, but I've represented these here as filling up the cell, and they're found throughout the cell. And it's really, we have to, if we're going to manipulate the cell and its functions, it comes back to manipulating the activity of these proteins and thinking about how we can modulate these. I have a small video clip here, which is really a schematic trying to represent the inside of a cell and the large number of proteins that are found in this cell. And this is really a very trying to show it and think about what's inside of the cell. We often think of a single molecule that we're working on, but it's really a full and busy place with a lot of proteins, and they are constantly running into each other, but most of those interactions result in nothing. And it requires the specific interaction of proteins which are supposed to interact, which results in activity. And we can see these busy proteins colliding here, and we are assembling a machine. And these kinds of protein assemblies are what controls the action of the cell. And they will assemble in response to a given signal and then result in specific activities. And we can see these busy proteins coming together, and then in this case, we're activating some molecules, which will then go on and chop up things. And this is really, you can think of these molecules as a bit like the hungry enzymes in your laundry detergents that I think that are commonly used in ads, and they go along and they munch up things. And so that's just to kind of to give you a picture of the busy place that a cell is. And I guess proteins are pretty complicated things. Where did my interest in proteins begin? Well, it began as a PhD student at Massey when I worked with Professor Ted Baker. This is a picture of him from about that time, and he had a group that included Heather Baker and Brian Anderson and Jill Norris, who's not here, who were long-standing, not shown here, long-standing members of his group. And then my project actually was a joint project in the early years, mostly supervised by Associate Professor John Tweedy. And I think nowadays we would call this a multidisciplinary project or some such thing, because it was really what I did in Ted's lab was to make and express and purify the first recombinant protein. So up until that time, the study of proteins had largely been limited, well, in the early years, to proteins that you could purify from natural sources. And so people would go to the abattoir or grow up a yeast culture and purify something, and so you were extracting proteins. And so really what I did was to make the first recombinant protein in the Baker laboratory. And so this required me to clone and overexpress my protein so I could analyze it. And at least at Massey, at this time, this wasn't such an easy process because we had no PCR machines. Now, most of our undergraduate students get to do PCR reactions and things, but I had to go to Ag Research to use the PCR machine. And then, in fact, I didn't, and nowadays many of the students who I see here, they would purify their proteins from E. coli. Actually, I set up a mammalian expression system, which was quite a challenge given that was our first efforts to make recombinant proteins. For that, I went to Vancouver for 2 months to establish the system. And then I came home and established that here. And this was a complicated expression system where we had to express the protein in the soluble media and recover it. Really not a trivial endeavor. And then we had to purify it. And I guess I kind of like the fact this PNGase F, the protein that that was secreted was glycosylated, and we had to remove the carbohydrate. And so we couldn't buy PNGase F at that time, which is a deglycosylating, an enzyme that will chop the carbohydrate off. Instead, we would grow big cultures of Flavobacterium meningocepticum, and we would purify it from that. And really, we didn't require nearly the compliance that I suspect we would need to do that nowadays. So once I had purified my protein, I crystallized it, and then I solved the structure of this protein. So this is half of the structure of human lactoferrin. So lactoferrin is found in secreted fluids. So it gets its name because it's very prevalent in human milk or mammalian milk. And really the great thing about being able to make recombinant proteins is that we could make mutants and analyze them. When you're purifying proteins from natural sources, you can't make a mutant, so you can't specifically investigate the role of a given amino acid. And so this was really the great thing about being able to make recombinant proteins. And so really the system I established in Ted's lab, a lot of mutants I made, and they formed actually the basis for a large number of projects. And I think there are 8 or 10 papers that resulted from that work because I effectively set up a system. So if we just take a few moments and look here, so what do we learn by solving the structure of a protein? So this, we learn a lot of different things, but we learn about topology. So when a protein is synthesized as a linear sequence, that's not the form it takes. It folds up so that specific groups come into close proximity, and we can see this here. So we have different elements of secondary structure that we would refer to, these arrows and these 12s and then these squiggly bits. So these therefore define the structure that we have. So we learn about topology. We also learn about the surface, and as I'll talk more about, a lot of my work has been focused on protein-protein interactions. So that's really how do two proteins bind to each other, and it's these surface properties that are important for that. We also learn a lot about the chemistry of the proteins and how things are bound. So lactoferrin is an iron-binding protein. That's its role in the it is to bind iron tightly and keep it away from bacteria effectively. And so in the structure I solved, we could see that the iron is coordinated here by these specific groups of atoms in a very specific arrangement. And then really when I made mutants, we can investigate the role of these different residues and see how they function. So these are some of the different things we learn by solving a protein, and we can represent our proteins in these different ways. And then sometimes it's just easiest to represent our protein as a nice little sphere, and you'll see a lot of these in my talk because if I show all of these big complicated structures, sometimes things are missed. So these are the different ways that you'll see me represent a protein. So as Helen indicated, after I left Massey, I went to Berkeley to work in the lab of Tom Alba. He was really a great supervisor, and I worked on proteins. I solved the structure of a domain involved in a protein involved in colon and inherited susceptibility to colon cancer. But I learned a number of things there, and perhaps one of the things, when I moved to Berkeley, he also had just moved to Berkeley. So I learned about setting up a lab because I had to buy the chairs and the glassware and measuring cylinders. So I might have done less work, but I think I learned quite a lot in his laboratory. And I also really where I developed my interest in cancer. And so for the rest of today, I'm really going to talk a lot about proteins involved in cancer and how they and how we can manipulate these for, I guess, for the benefit of for our benefit. So just to step back a moment, when we think of cancer, if we think of this as just a representation of a monolayer of cells, and on this side we've got normal cell division occurring, and on this side we've got some cell division that's representing what might be occurring in cancer. And you can see here that these ones form a nice layer. Nothing's they're all just nestling up against each other. Things are looking quite orderly. Here things are starting to go out of control. And this is really what happens when a tumor is forming. And so this is really what we think of as tumor formation. And so this really then tells us that there's a critical balance between cell division and cell death. And so it's amazing that we don't get more diseases, because this has really got to be a tightly controlled process. And on this video clip that we just saw on the left, our cell division's being exactly balanced by cell death. And so you get this perfect balance of number. And really for most of us, once we're adults, if all's going well, we stay roughly the same size and we have roughly the same number of cells. And so this is really a really exquisitely balanced process. And if we get an imbalance between division and death, or apoptosis and proliferation, I'm probably going to use apoptosis quite frequently, really that means cell death, then we get various disorders. So when we've got, the fact that a cell becomes damaged is not in and of itself a problem, because normally there are lots of pathways to remove those damaged cells, but if they're not removed, we get this accumulation or imbalance of cell number. And that's, as we have just seen, what often occurs in a cancer state. We also have disorders associated with too much cell death, and this is really what happens in a lot of neurological conditions. So a lot of our neurons should last our whole lifetime. If they die prematurely, that's not what we want to occur. So this regulation of cells of survival is critical. And so apoptosis or cell death is not just about protecting us from disease. It's also a critical process for development. Some of the things that are, I guess if you disrupt this process in mice, some of the things that you see this webbing rather than nice little digits, and for the sculpting of organs. So it's really a critical process that occurs every day. And in each of us, billions of cells are dying each day by apoptosis. And this is really a highly controlled process. And we know a lot about this process because Sidney Brenner, Bob Horvitz, and John Sulston received the Nobel Prize in 2002 for their discoveries concerning the genetic regulation of organ development and programmed cell death. So really their work laid the foundation for our understanding about how cells die. And we can represent a lot of what they know, what they discovered here. And really this is also a fabulous example of I guess the beauty of basic biology. So by studying the little worm, this little flatworm C. elegans, they were able, so Sidney Brenner really established this worm or his laboratory as a model organism. And then John Sulston, he mapped the fate of every cell, and kind of remarkably, he showed that cells don't just die randomly. It's really a very controlled process. And therefore, because it was a controlled process, he was then able to figure out and identify specific molecules which controlled this process. And so then these molecules were cloned in Bob Horvitz's lab at MIT. And so we have these different proteins, and I'm going to use the same color code throughout. So we have these proteins here, Eagle-1, which inhibit. So these arrows, well, these not arrows, these connections like this indicate inhibition. And then these, when I use an arrow, that indicates activation. So actually in a healthy cell, normally this protein is dominant and it inhibits cell death by inhibiting this protein. Once we get this guy, we get 2 inhibitors of the inhibitor, and so you activate cell death. So I'm afraid it's somewhat complicated, but I'll try and keep it simple. Anyway, or as simple as I can. So the other remarkable thing is that this process in worms is actually very conserved in us, and we have got homologs of these same proteins. And in fact, if you take the human protein and put it into a worm where this protein has been knocked out, it complements. So the function of these proteins is remarkably conserved. And in humans, this homolog is called BCL2, or B-cell lymphoma protein 2. And so this was first identified at a translocation breakpoint, so as a genetic change which is found in B-cell lymphomas. And this really then laid the connection between apoptosis and its importance as effectively a hallmark of cancer. And so if we go to the next thing, when we think of the hallmarks of cancer, there's this very classic paper in 2000, and there's been an update, but really it remains a sort of a landmark in the field. And one of the hallmarks of tumor formation is this resistance to cell death. So the cells which should normally die don't die, and so this, I guess, a number of connections led to this. And this really then identified, and there's been a huge effort to develop small molecule compounds, or what would be commonly implemented as drugs, which can manipulate this process and trigger cell death, because the desire is to trigger cell death in the tumor cells so that we can eradicate these cells. So what have I done? So really, I'm going to tell 2 short stories. And what I'm going to focus on is really kind of the minutiae, because that's actually what I do, I'm afraid. It's picking a target protein and then figuring out how these proteins interact and how then we can modulate and develop small molecule compounds which will affect the activity, remembering that we've got our cell full of molecules, and we've got to develop compounds which will specifically bind to the right protein and modulate the function of that. And then if we can develop these compounds, then maybe they can be turned into therapeutics. And then in the second story, I'll really talk a little bit about a compound where we've learned a lot about the function of the second story, the compound is in the clinical trials at the moment, but it's also told us a lot about biology. And so sometimes these are iterative processes. You shouldn't just think of them as an endpoint that you're going after a drug, because many failed drugs are very good agents for manipulating signaling pathways. Okay, so we're going to focus in on this pathway which I've shown you already, and we're going to look at these proteins up here. So we're going to look at our friends, the BCL2 proteins. These are our pro-survival proteins. And then what I'm going to refer to as the BH3 only, or you can think of them as the yellow death proteins. And so these proteins, in C. elegans, there's just one. So we've got this simple interaction that's a one-to-one correspondence. In humans, we have, or in all mammals, we've got homologs, and so we've got families of proteins which serve similar functions. And really, there's been a lot, because we know that these proteins can inactivate these proteins. This interaction was really a major focus, because it was thought that we could develop compounds which would inhibit, by mimicking the activity of these proteins, we could take these guys out, and this would allow us to develop improved therapeutics. And I think really this holds true, and there are a number of companies that have exactly these mimetic compounds in clinical trials. So I'm going to sort of take us through a couple of the things that we have done which have helped with this along the way. Okay. So I'm a protein biochemist, so that means we look at proteins. And I am going to talk about 2 different kinds of ways that structures can be solved. And most of these BCL2 structures that we solve was solved actually by NMR. And this was all done in collaboration with people in Melbourne, and actually using the Bio21 facility there to collect all of our NMR data. And really, this is a complicated jigsaw is how you can think of solving a structure. And so you collect a lot of dots, and then these dots tell us about which atoms are in close proximity. And then really by spending months at the computer, you get a big list of these dots, and then you can turn them into a structure one day. So it's a lot of kind of jigsaw building. And so some of these structures, this one actually was a relatively straightforward one, but some of them took really 6 to 8 months to solve. So this, going from here to here is not a trivial process, but really what does it tell us? Well, the key thing that these structures told us by looking at different members of this family, so we're looking at our pro-survival blue guys, is that the conserved residues, so these proteins have 3 groups of residues which identify them as being a member of that family, and they actually come together in space. So although they're distant in sequence, once the protein is all folded up, they come together, and we can see them here associating, and we'll come back to these in a moment. So we also looked at these BH3 only, so our yellow death guys. So these ones, in fact, we couldn't get a pretty picture like I've just shown you. These proteins are actually unstructured in solution, and this was really one of the first classes of proteins that was quite well studied as being an intrinsically disordered, and so this is a CD spectrum, and for most of us it doesn't matter too much, but the key thing is that these are really the kind of the spaghetti proteins. They're not well defined, they don't make one of these pretty structures. But then what we did is we solved a number of different complex structures. And when I say we, most of this actually, the structural work that I'm showing here was done by Mark Hines and I together. And this is one where the first turnaround was the protein on its own, and then it binds to this yellow one. So this is our yellow death protein, which is coming from here. And so on its own, it's a piece of spaghetti, and we could just draw it as a squiggly sort of twirl, and then upon binding it becomes ordered and it adopts a very discrete structure. And all of these BH3 only proteins, the region that binds adopts exactly the same structure, at least when looking at it at this level. Likewise, all of our pro-survival proteins, our blue ones, now all of these family members have been solved, and they also, at this kind of topographical view, have the same structure. And so this told us a lot about the conformational changes. We also solved another structure, and this is another pro-survival one, BCLW. And then this is the most complete. This was the full-length protein we solved. The rest of them actually had their C-terminal tail missing. And these tail residues, so this can sort of be the tail of the dog lying down here, it's actually lies in the position where our yellow protein binds. So if you look here, we've got our green, pink, and yellow. And we actually showed that this tail modulates the ability of proteins to interact with this conserved hydrophobic groove. So we can kind of bring this all together, and if the last bits have kind of been leaving you cold, well, maybe this is kind of a summary of what we've just seen. So we've got our spaghetti protein, we've got our pro-survival protein, which really keeps its binding groove kind of occluded, which makes sense if it wants to prevent non-specifically bind to proteins. And then when they come together, you get this complex. And so at the same time, these proteins tend to insert into the membrane, and so we get this complex forming. So how are we going to develop a drug? What have we done to help think about making a therapeutic compound? Well, in parallel with these experiments, we started to wonder if some of the family members were slightly different. And in fact, together with David Huang at the Walter and Eliza Hall, my group purified all these proteins, and then we used them to just do a very detailed analysis of the binding properties. And what we showed is that these are our 5 pro-survival proteins and our 8 yellow pro-death ones. And the height of this bar indicates binding affinity. So how tightly do they interact? And we can see, this is a log scale up here, in fact, so there really are big differences in our binding affinity. But if we look at our red ones, it actually binds tightly to all of them, so the bars are about the same height. But some of them didn't bind equally. And we can see this here, for example, Noxa. only binds to these two proteins, A1 and MCL1. In contrast, this one, bad, these have terrible names. So this is kind of a good protein, but it's got a name bad. It only binds to these guys. So these proteins don't behave exactly the same. So they actually have selective binding. And so why do we care? This paper, which has actually been cited over 800 times, so really a highly cited paper. And so why is that? So actually it's because the selective binding matters for killing. So if you want to kill a cell, you have to take out all of the pro-survival proteins, not just one. And so if you made a bad mimetic, that could be a good mimetic, but if it's mimicking the action of this protein, and if the cell has these other proteins present, it won't kill it. And so this is really critical if you're going to make mimetic protein compounds that mimic the action of these. They've really got to take out all of the pro-survival. So we tried to understand, so we also did some, this is the work of Callan Smits, my first PhD student. He did a huge amount of work in my lab, and I should say these were some of the first structures I think solved from the synchrotron in Australia, in Otago. And so he solved 4 different structures, so these are complexes of these guys. And what we were looking for is looking for differences in affinity. So this is actually one that's relatively weak. These are greater than 200-fold difference in affinity. At this level, they look the same. So it's not until you start really drilling down and looking at the detailed context as to what might differ. And we can see in this one, this interaction. So these other 3 which bind tightly all have this set of dots across here. And so these ones are missing. And so it's these kinds of differences which underpin the selective binding and give rise to differences in affinity. And we can, so in parallel with these experiments, so WeHi has a high throughput screening facility, which I think some of you will have heard about before. And we use some of our recombinant proteins in the high throughput screen. So we put them through this high throughput screen where we're searching for small molecule compounds which would bind tightly. And then these might be lead compounds for further development as drugs. And so we were doing these experiments and we came up with some compounds that were binding. But then really in 2005, there was a beautiful paper from Abbott Laboratories. And so they came up with this compound, which is now, I guess, one of the leading compounds of this class, referred to as Abbott 737. And so this compound has many of the features of what you would want in a mimetic of these yellow. So we're wanting to mimic the action of these yellow proteins with a small molecule compound which has features which might be able to be delivered as a drug. So you might say, why not just deliver this protein itself? But if you gave someone a protein, they would actually think it was food and they'd probably chomp it up and use it as energy. That would be often the response to giving people protein. So they're not always a good therapeutic. So if we can, the goal is then to come up with these small molecule compounds. So Abbott came up with this compound which looked very good, and it led to regression of solid tumours, and then they were also able to show in preclinical data that it was well tolerated and I guess efficacious in treatment of certain tumours. And so what came from this? So really then the analysis of this compound, which is advancing through clinical trials, was significantly aided by our knowledge about selective binding. Because it turns out that ABT 737 is a mimic of this protein bad. So actually it doesn't bind to MCL1, it doesn't bind to A1. This means it doesn't work in tumours that have got these proteins present. And so then the knowledge about our binding profile has been really significant in advancing that compound through. Because if we hadn't have known that's how it worked, then actually the compound would look much worse, because unless you can type the tuners and find the right tumors that you can then analyze the compounds in, things will look like they're failing, but it's really a matter, and I guess it's this kind of analysis is what you can think of as some of this personalised medicine, which one day we all hope will be developed and help us before we need it. And so this compound then is being used as in clinical trials, but they must type the tumours to make sure that they haven't got MCL1. This is one of the early experiments. This was work that David Huang published, and we can see here, this is looking at mice with lymphoma, and we can see this is a tumour which is expressing MCL1. These mice die quite readily soon after 20 days, whereas when you treat the control or the same tumour which has been engineered to express BCL2, then this tumour is killed and the mice respond to the therapeutic. So this is really quite a promising avenue for the development, and I think that there will be, these will advance through to the clinic, or at least analogues of them. So just to recap this story, in a normal cell, what needs to happen? So in a normal cell, we get lots of stresses, and the typical stress on a cell causes a signaling pathway which leads to our yellow protein, which says, okay, there's trouble in this cell, we need to die. So this yellow protein gets made, it then binds to our blue protein and signals apoptosis and death. So that's what we want to happen when a cell becomes damaged. For example, if we get sunburnt, we want our sunburnt cells to go away. We don't, but in a tumour cell, the common thing that happened is we've got disruption of the signaling pathways. So rather than responding appropriately, there's a blockade. And so although there's a stress and the stress might be perceived, you don't get, there's no ability to increase the expression or make the yellow protein available. And so these cells can then survive, and then this might then go on to replicate and give rise to a tumour. Whereas if we can give our BH3 compound, so if we've got our mutant signaling pathway, then we can then come in with this compound kind of down right at the end. And these compounds are being, one of the things that's just looking quite promising to co-administer with chemotherapy and radiotherapy, because a lot of the standard treatments, you create a stress, but if the signaling pathway is broken, the cell can't do anything. It can register the stress, but it can't go on to connect and say, okay, we need to die. So this then, if we can make this mimetic compound, which is now we can, things are looking more promising. So I think whether these compounds that are in the trials now progress or whether there'll be different iterations of them, time will tell, but they at least look quite promising. Okay, so I want to thank a number of people for this story, so particularly Mark Hines and David Huang at the Walter and Eliza Hall Institute. So I've worked with Mark for a long time before and after that project, and David particularly for about the 5 or 6 years when we did this work. They were both excellent collaborators. Okay, so the other thing, I'll just take a small break before we do a shorter story, so don't worry. I just want to also make a few comments about equipment. So when I came to Otago, there was relatively a small amount of equipment for doing protein research, and I really think that the investment in equipment has made a huge difference. So we have these boxes, and I think I've heard Peter Crampton refer to biochemistry's boxes. This is a $1 million box. So these are valuable and important boxes for research. So that's was funded by the mass spectrometer that was funded by the Large Equipment Fund. We have our X-ray facility, which was particularly strengthened by the arrival of Kraus, and then has been a major resource for a number of other people. And we have other pieces of equipment which have been funded by the school and the division, and these have all made a significant contribution. And now I think biochemistry is really a good place to do protein research. And so there's also a number of people that lead groups which utilize this resource. So John Cutfield, Sigurd, and Torsten. And really, I think John, and when I first came, we had to travel, and I know that John and Sue for a number of years had to travel enormously, and it really, being able to do things here has made a huge difference. So I think the investment in equipment has really allowed a lot with what we do. Okay, so now we just want a short story to finish with because we'll be running out of puff. So this is really where I want to talk more about a small molecule compound which is also being in the clinic for the treatment of cancer, but it's really also told us a lot about how the protein works and then really sort of identified other avenues for treatment or development of compounds. So we come back to our pathway that I've talked about and we've been focusing up here. Now I'm going to talk about these green guys down here. So these are called IAPs. So IAPs are inhibitors of apoptosis. They bind and inhibit caspases. And in that very early clip that I showed you of proteins running around munching things up, the proteins that were doing the munching were our caspases. So their job in the cell, once the apoptosis has been activated, is to get about and destroy things. And so you really don't want to let those guys loose unless you really want to die. So this IAP is really the safety catch. So these bind and inhibit the caspases and really prevent latent level or non-specific activation. And then I'm afraid these inhibitors are regulated by some more inhibitors. And so as we saw before, we've got kind of this complex pathway. So these are our IAPs, and they're fitting in here. And I'll come back to this slide and thinking about how they work. But what have I got next? Okay, so this is a collaboration with David Vo and John Silk, and I really have to thank David Vo enormously. When I came back to New Zealand in 1997, somehow I had an email from him early on within the first week or two, and so really then that's when I started working on IAPs, and I have worked on them on and off ever since. And then he also, really all of the WeHi collaborations have been I guess, helped by David Vo. And he's a great person, and I can always ring him up and he'll give you at least blunt advice, if not sage advice. And he's now the Deputy Director of the Walter and Eliza Hall Institute, so he's very good. Okay, so IAPs, the IAPs that I care about are multi-domain proteins that look like this. So they have a bunch of domains, and so this means, in fact, and they have been relatively tricky to study, and so we've had to focus on different domains. And in the early days, I focused on these domains here, baculoviral IAP repeat. So not an easy name, but this is the defining domain of this family, and the name tells us quite a bit. These domains were actually first found in baculoviruses. So again, we start with basic biology, and that then has come to sort of have important ramifications for how we might treat human disease. So this is actually baculoviruses infecting an insect cell, and that's where they were first found, and then subsequently David Vo's group, as well as Dave Goodell's group at Genentech, found the mammalian homologs, which I'm showing here. So they all IAP proteins have a BIR domain, and Mark Hines and I solved the first structure of a BIR domain back in 1999. It's a fairly harmless looking domain. It's got a little zinc ion here, and then it's 100 amino acids, fairly, and at the time we didn't know a lot about how it worked. But really, we didn't do a lot more on it for a number of years, but lots of other groups figured some really important things out, and it really so our BIR domain here became quite an important drug target. What we have here is, it's just showing IAPs, but we can think at this point of the BIR domain as representing the IAP. And this binds to a protein called SMAC. And the region of SMAC that binds that's important for the action is just 5 amino acids. And this complex is shown here. So when this protein SMAC binds here, this then means that IAPs can no longer inhibit caspases, and so you activate the caspases and you signal apoptosis. So that's a good thing if you want to kill a tumor cell. So this meant that it was really a beautiful target for drug discovery, and lots of companies, so there's a whole list of companies that have IAP antagonists, about 6 or 7. And so this is a small molecule compound. So this is a little chemical that we might be able to use as a drug, binds in exactly the same spot here. So when this chemical binds, the idea was that then you would block this bar here, and so then you would get an active caspase that could go around and eat up the cell. So that was all very good. And then this is a figure from a paper of John Silk and David Bose, and this is really a landmark paper in the field because the compounds do something really remarkable. They bind here, and they were expected just to activate caspases by relieving the inhibitor. But in fact, what they do is they destroy IAPs. So if we look here at this is showing from extracts, and we look at this IAP levels in cells, and then 5 minutes after adding, so the people we work with at Tetralogics, their good compound we call compound A. So it's the good one. And after 5 minutes, you can see that remarkably, this protein has gone, completely wiped out. So what this means is the compound binds here, and we've got structures, and we know how it binds, but it does something completely different because just binding here, really you wouldn't expect it to destroy the protein. So this really brings us to focus on the ring domain, and this has really been a discovery over the last 5 years to try and figure out how binding here, in fact, activates the ring domain. The loss of protein, so this rapid loss, means we have to think about ubiquitin. So ubiquitin is a marvelous protein if you want to destroy things. So it's a very small protein that can be attached to other proteins, and when it's attached to them, it says that they have to be sent to the rubbish. So if we've got our green protein, we stick a few ubiquitins on it, and then, off we go in to get recycled. And so I guess it's the environmentally friendly protein or process, and so we keep our ubiquitin and we get to reuse it. But that's the process which is resulting in the loss of that band. So you go from having that band to its loss because we've sent it off to get recycled. And we know a lot about this process by some really beautiful work of Aaron Chechanova, Avram Hershko, Irwin Rose, who really, over a long period of time, he was the PhD, or Aaron was Avram's PhD student, and these two still go to many of the ubiquitin meetings, and they're very helpful people. But they worked at the Technion Institute in Israel, and they used to go every summer to Irwin Rose's group in the US and did their pioneering experiments effectively over a number of summers. So pretty impressive. And they really discovered ubiquitin-mediated protein degradation. And so it's a really complicated process, I'm afraid, and so we really don't want to, it's getting too late to think about that. But we've got a number of proteins, and really our IAPs, because they've got this red domain, means that they fit in here at this slot. And there are actually 500 of these proteins, and we really know very little about any of them. So lots of these proteins, ubiquitin has really it's really not a well-studied thing, and so I think this is an area where lots of things can be discovered in the future. But this presence of this domain tells us that it belongs in this slot as an E3. So that means it can stick ubiquitin onto itself or onto targets. And so all of these other 500 E3s will have specific targets that they're going to send to the recycling bin. So when they're working, they're going to cause these proteins to go and get recycled. So we were interested in the ring domain, and we could cause this, we solved the structure of it, we showed it was a dimer, and then, I'll just go reasonably quickly now, we then could show that dimerization is important for activity. So when you stick ubiquitin on, in a cell you send it to the recycling bin and it vanishes. If you do this in a test tube in my lab, you'll make a ladder, and if you run it on a gel. So this is really our protein with ubiquitin stuck on it. And we can see that it needs to be a dimer. Monomers don't really do the business. So dimerization is important. We then showed that dimerization is regulated, and some work with Pascal Meyer in London last year. We could see that other domains, so if you just take the ring domain, it's always a dimer. If you take the full-length protein, we could show that this is regulated and that the N-terminal domains modulate dimerization. And that the presence of the dimer correlated with activity, So when we've got a dimer, we get a much bigger smear than if we've got a wild-type protein, which is forming little dimer. Okay, so in a healthy cell, I guess, this is normally a monomer, and these domains interact, and this prevents activity. If we make it into a dimer, we know that then it can interact with the ubiquitin machinery and start sticking ubiquitin on. So now if we come back to our compound, how might this be regulating this pathway? So it's really quite, I guess, it's pretty cool. And so what we figured out is that these compounds, so this is our magic compound A. So this is really a good compound. It's in clinical trials and looking pretty good. So when we add this compound, we can activate our protein to make a much bigger smear. So this is in a test tube, we see it as a smear, but if you add it to a cell, you knock out that protein, so we see the loss of our band. And this correlates exactly with it forming a dimer. And so compounds that activate this promote dimerization. And so I guess I've gone through that quite quickly, but if we just take a few seconds on, or a little bit of time on this slide, it really summarizes how these compounds work. So these compounds can bind to the BIR domain. And they were really meant to do something very simple, just to knock off a caspase and activate it. And that's not what it does. It does something much more complicated and perhaps much more interesting. So when the compound binds, it triggers this protein, so our red domains now interact and form a dimer. This means we can interact with the ubiquitin machinery, which means we can put ubiquitin on, we can then send it to the recycling bin, and then this is why we're going to have this loss of this protein. And it's this loss of this band which is what you're looking for if you want to kill a cell, because these proteins are potent survival molecules. And these are, I guess, these are at least a very promising avenue. And so just to come back now to finish, really what I've talked about today is two short stories that kind of connect together on this apoptotic pathway. And so the first story we talked about was developing mimics of these molecules so that we can activate or inactivate our pro-survival and promote death by that way. And then the second class of compounds are IAP antagonists. Take these guys out of action, so you activate caspases. Actually, it turns out, and I've tried to keep it simple for tonight, primarily we're actually knocking out these IAPs up here, and we lead to a whole new cascade. We activate transcription factors, NF-kappa B, and it has really a dramatic effect on the cell signaling. So we've learned a lot about cell signaling from this study, and so I guess I think I've covered that, but not only are the small molecules leading to therapeutics, but they're really telling us a lot about the cell, and I think that's really what you want to be doing, because lots of small molecules can fail. So hopefully if you can utilize that knowledge and not just go after a drug. Okay, so really the goal of a lot of this work is really to try and take out this resisting cell death so that you can therefore kill the tumor cells, and then I guess this is the goal of many, many groups around the world. And the story that I've told, I hope, just gives an idea about what a relatively small group can do, but you can think we've made quite a contribution to our thinking about the development of these compounds and how to, I guess, manipulate cell signaling pathways. So these people here all need a lot of thanks, because they have all remained good friends throughout my career, and I've keep in touch with these people often, and so they have, I think I've mentioned their contributions already. I also want to thank particularly Warren. So I came to Otago when Warren was head of department, and he was an enthusiastic supporter and helped with many things in terms of writing grants and getting money for equipment. And again, John was helpful in establishing, and I think we owe John a lot for helping to set up protein research in Otago. And then in the last few years, as Helen has noted, I've done some work as Associate Dean of Research, and she's been very supportive. Now, I really need to and so I decided not to put pictures of people throughout the slides, because I thought that then that's only representing the people that I've talked about, and really their work, which is just a snippet, and there are a lot of other people involved. A lot of the people who were previously in my group are shown here, and I want to particularly pick out Peter Mace and Callum Smitch, who made a huge contribution to the projects that I've shown, but a number of other people here. And a lot of the early work was particularly involved a number of excellent technicians, and Raylene Frederick Short, Penny Harrison, Gretchen Ski were also all technicians, and then Bodie Betjeman, a more recent technician who just left to be a winemaker, alas. So he's gone to the good life. So I really want to thank the contribution, and really my lab has mostly been quite a small lab. And these are the current people in my group, and actually I haven't shown any of their work. We mostly work on ubiquitin-related processes, and I think there are a lot of exciting proteins, so we're really enthusiastic to understand the process, and there are actually very few drugs that manipulate the ubiquitin pathway, and so I think there's a lot of opportunity there in the long term. This is my lab from a couple of years ago, and many of these people are still in my lab, and so they're a really good group of people to work with. And I think I've mentioned a couple of these people, but Callum and Peter really made a huge contribution to this work. And then I wanted to talk about Jo just for a moment. She's now in Australia. She was a very good PhD student, and she worked on a project, and it was really in many ways the same, but in that we were doing a very careful analysis of a protein. But in this case, we proved that the literature was all wrong, and so her work led to retraction of an article from Nature and things. So it showed the perseverance, or the value of persevering, and so she did a great lot of work, but in this case it was a slightly different outcome. So lastly, I really have to thank the Marston Fund. They've funded me consistently since 1997. When I first wrote a Marsden grant, it was, I think, in days when life was easier, and I was very naive. I got the referees' reports, and I thought, oh, yep, those comments are fair, and I never wrote the rebuttal because I thought the comments were good. They were reasonable, and so but they still funded me, and so I think, and then they have really been a fantastic support of my group. I've had HRC funding, which I have at the moment, Oncology Trust, Lottery Grants Board, and then the university in numerous ways has supported through specific grants, and then equipment, and health sciences postdoc fellowship, which was supported part of Peter Mace's as a postdoc. So thank you. And then I just wanted to thank my family. So nobody wanted, well, Richard didn't want a big picture, so we went with this picture as representative. He thought that you couldn't pick out anybody from looking at this. So I wanted, I guess, to thank Richard for all of his support, and then my whole family for various aspects and helping in all kinds of ways. And thank you again, and hopefully I haven't gone too long. So thank you very much.
Catherine, thank you very much for that outstanding lecture.
Go out the door now.
And we all enjoyed it very, very much. My name's Kraus. I'm the current head of Department of Biochemistry, and it's my privilege to say just a few words about your lecture. I can't add anything to it scientifically because it was such an excellent job, but I thought in so many ways that the lecture and that your career really typifies outstanding science, and I thought it would be good to review some of the aspects of that, and particularly for some of the young scientists in the audience who are wanting to construct a career as a kind of a guideline about how to go about doing it, because I think you've done it and continue to do it so incredibly well. The first key aspect is that you chose a very, very important project. My PhD advisor used to always talk about choosing a project that you could chew on over and over for many, many years. And I think you chose an important project on two different levels. One is on the very basic science level, where all the proteins are moving around inside the cell, and how these proteins interact and how they find their partners in order to tell the cell, okay, you need to die, you need to live, and that molecular ballet, if you will, is incredibly neat from a basic science point of view. But the area that you chose to focus on is also incredibly important in terms of translation because it underpins treatments for cancer. So it's important on a basic science level, and it's important on a translational level, and it's something that can be worked on for many, many years. The second key thing I think you've done is you've consistently chosen outstanding collaborators. And these are people who are on the cutting edge of science, who are moving science ahead, and you mastered the literature in the field and mastered the knowledge and worked with these people and continue to produce outstanding work year after year after year. The third thing is you're trained as a structural biologist or a protein crystallographer, but you moved over to the dark side and did NMR structures, right? And I think that the hallmark of that is you were pursuing a scientific problem. You weren't pursuing a technique. And it's easy as a young scientist to fall in love with whatever technique that you're using at that given moment. But in fact, what we really want to do is to find an answer to a problem and to use whatever techniques it takes to find the answer to that problem, if it's NMR or crystallography or genetics or what have you. The fourth aspect of your career that I think is really, really remarkable, and you mentioned it yourself, and that is perseverance. And perseverance is so important because science is a hard yaka. 90% of whatever we do fails, and it fails again and again and again, and you have to stick with it if you're ever going to succeed. And finding out that the ring domains were dimers in order to be active took perseverance and incredible strength in order to report that, and has ended up changing the field. Then the fifth aspect is particularly important, I think, in a time where people worry about how to fund science, and should you fund translational science, in other words, science directly reporting it, to a clinical result or an agricultural result, or should you fund basic science, which is looking at a basic problem? You can see that as you've worked in this field over time, the translational or the clinical compounds have come. That wasn't the initial, that may have been the initial goal, but that wasn't what you were initially working on. But now Catherine has collaborations with pharmaceutical companies around the world, and they're coming to her to test these compounds. And it's really hugely illustrative about how if you choose an important problem in science that has clinical ramifications, those will come eventually. But it's very, very difficult to fund that at the beginning. So by funding basic science, you end up getting things that help all of us, but a little bit down the road as opposed to right away. So I wanted to end by saying thank you very much for this brilliant example of outstanding science. We're very grateful. We're grateful to have you in biochemistry. We're grateful to have you at the University of Otago. And we look forward to continued discoveries from your lab for many years to come. And I have a small token to commemorate your inaugural professorial lectureship that I'd like to present to you.
Thank you.